Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, I hope all of you are fine and safe at your home.
Dear students, the learning goals for today's lecture are we will study about the host organisms which are used to incorporate the cloning vectors. Then we will study about the restriction endonucleases, their types, what is the mode of action of restriction endonucleases, and we will see the example with the DNA fragment and the cloning vector which are treated with the same restriction enzymes. Hosts for cloning vectors. Various kinds of hosts are used by different cloning vectors, like we can select the host depending upon the various properties. Like we need rapid growth, so the ideal host should be able to rapidly grow in inexpensive medium, because we want to be economical in lab, so we want the more growth of the host cell by using the inexpensive medium. The second important characteristic for the host is it should be non-pathogenic. We know that pathogenic and non-pathogenic bacteria exist, but we make non-pathogenic hosts, and mostly the hosts which are used in the research in biotechnology or the molecular biology, these are disarmed non-pathogenic strains. Some part which causes the pathogenicity, that part is removed from those host organisms. The third important property is the host organism should be capable of incorporating the DNA. Then it should be genetically stable within the culture, and it contains appropriate enzymes that allow the replication of the vector. For instance, they contain the DNA polymerase enzyme. Remember, in the vectors property, we have discussed about the presence of the Orici locus, and Orici locus will work only when the enzyme is present. Enzyme, which is DNA polymerase, which will recognize this Orici locus, and then it will start the process of replication of the vector. So, a vector should possess, a host should possess these five properties, which are the growth in inexpensive medium, it should be non-pathogenic, it should be able to incorporate the foreign DNA, it should be genetically stable within the culture, and it should be equipped with appropriate enzymes that allow the replication of the vector within the host cell. Most commonly used host organisms are bacteria and eukaryotes, which are used for cloning purpose. For the expression purpose, we can use insects also, we can use mammalian cells also, and we will discuss about the host organisms in detail when we are done with the cloning, and we will move towards the expression of the protein within the expression system. So now our focus is on the cloning host, and the cloning host which are most commonly used, these include the two bacteria which are the E. coli or the Bacillus subtilis, and one eukaryotic organism which is used as a cloning host, it is yeast, which is Saccharomyces cerevisiae. So the advantage and disadvantages of these cloning hosts are shown in different colors. Advantages are represented in blue color box, and the disadvantages are in pink color box. So the advantage of E. coli is that its genetics is well developed. We know about the genetics of the E. coli, on the basis of which many strains have been produced. So many strains of the E. coli are available. I will give you an example of a strain which is used for cloning purpose, and it is E. coli strain which is DH5 alpha. Similarly, many different strains have been developed. One is DH10 alpha, and so many. This is the best known bacterium and most commonly used host. Its disadvantage is that it contains certain the traps proteins, which are the channel proteins, which are potentially pathogenic, and these proteins are actually used for the movement of the compounds because they are the channels. They are used for the movement of molecules inside the bacterial cell, which could be pathogenic for our gene of interest. Bacillus subtilis, it is easily transformed host, it is non-pathogenic, it naturally secretes the proteins which are being expressed in it, and it is actually the endospore formation and which simplifies the culture. It means that the culturing of the Bacillus subtilis is easy. But its disadvantage is that it is genetically unstable, that it is not able to carry the vector in it for a longer time, and the vector will get lost from it, or it will lose the vector with the passage of time, and it is genetically less developed in comparison to the E. coli. For the proteins of the eukaryotic organisms, we know that there is a difference in the gene structure of prokaryotes and eukaryotes. And if we are working on the eukaryotic genes which are post-translationally modified and they contain certain properties which cannot be produced within the bacteria, then we have to move towards the eukaryotic host organism. So for that purpose, the model organism is Saccharomyces cerevisiae, and this has well-developed genetics. It is non-pathogenic. It can process the messenger RNA and proteins, and it is easy to grow. But its disadvantage is that it is unstable. It will not replicate most bacterial plasmids, because if we are introducing our gene into the vector, plasmid vector, and then we are using the eukaryote as a host organism, then it will be unstable, that it will not carry the plasmid. If we have to express our protein into the yeast, then we need to use the BAC or the YAC vectors, which are the advanced forms of the vectors that we have discussed in our previous lecture. So, on what basis we choose a vector and we choose the host organisms? There are several important characters. For instance, we should know about the insert size. What is the insert size? On the basis of the insert size, we use the vector. As you can see, all of these vectors, their host organism is E. coli, that they can be cloned into the E. coli. And if we are using the YAC vector, then we have to use the Saccharomyces cerevisiae host. YAC and the BAC, they can incorporate the vectors, plasmid vectors. This can be incorporated into the E. coli and the yeast host, but the most preferable host for the BAC is E. coli, and for the YAC it is Saccharomyces cerevisiae. And here the summary of the insert size has also been provided. So the choice of the vector, it depends upon the insert size. What is the size of the fragment that you are going to clone? So if it is 2 kb for instance, or 5 kb for instance, then you will have to use the lambda phage. If it is 2 kb or less, then you can use just the plasmid vector, which is not shown over here. Then important property is the vector size. What is the size of the vector that you are going to use? These are the vectors and what is their size. Then what restriction sites you are going to use? Either the restriction sites which you are going to use, they make a cut within your gene of interest or not. So you will choose the vector on the basis of the restriction sites that should be unique to your sequence. Then copy number. For cloning purpose, we use the vector which should be high copy number. High copy number means more copies of the vector should be present within the cell, within the host genome. Then what is the cloning efficiency of the vector? If the size of the fragment is small, then its efficiency will be high, and it should be It should be compatible within the host organism which we are going to use. Then the vector has the ability to screen for inserts. It means that the vector should contain certain markers that can be used to screen those vectors which contain the recombinant DNA. For instance, as we have discussed about the ampicillin resistance gene or the neomycin resistance gene or the chloramphenicol resistance gene, or we can use the double screening by using the LAC-Z prime gene, that we can insert a sequence in between that. Now we are moving towards the restriction endonucleases, which are important enzymes. Restriction endonucleases are the enzymes that cut the DNA at or near the specific recognition nucleotide sequence, which are known as the restriction site, which means that restriction enzymes will make a cut in a DNA sequence, and the sequence where they will make a cut, that sequence will be known as the restriction site. These enzymes can cut at the specific recognition site, as well as they can cut at the site near the recognition site. We will discuss when about the types of the restriction enzymes, then it will become clear to you. A special class of enzymes is the endonucleases, because different classes of nucleases are present, which include exonucleases as well as the endonucleases. So endonucleases are special class of enzyme that cut the DNA at specific unique internal location along its length. For instance, if it is double-stranded DNA, it will make a cut inside the sequence. It will make internal cut within the sequence. Therefore, we call them as the endonucleases because they cut within the molecule. These restriction endonucleases were discovered in late 1970s by Werner Arbor, Hamilton Smith and Daniel Nathan when they were studying about the bacteria, because these enzymes have been isolated from bacteria and they provide protection to the bacteria. These enzymes cut the foreign particle, like we know that the viruses, they infect bacteria very often. For instance, we have studied about the bacteriophages that infect the bacteria, and these viruses, when they are injected into the bacteria or they infect the bacteria, these endonucleases enzyme, they cut the genome of the viruses and protect the bacteria from these viruses, and they have received the Nobel Prize in 1978 for physiology or medicine for their discovery of these restriction endonucleases enzymes. They were the first one who have characterized and isolated the first type 2 restriction enzyme, which was the HIND HIND2. HIND2 enzyme was the first enzyme which was isolated and characterized from the bacteria Haemophilus influenza. Then it is become an essential tool for recombinant DNA technology. As I have told you that it is naturally produced by the bacteria and they use them as their defense mechanism against the viral infection. They chop up the viral nucleic acid and they protect the bacteria by hydrolyzing the phage DNA which infect them. So in this diagram, you can see that these are the fragment of the DNA which is cut with the EcoR1 enzyme. This black is the enzyme, which are the restriction endonucleases, and when it make a cut in this fragment it will cut, and in this fragment it will make a cut, then such ends are produced, which are actually the overhangs. One strand is longer than the other, and these overhangs are known as the sticky ends, and these sticky ends can be joined together to produce the recombinant DNA molecule. There will be a question that come to your mind that why the name restriction enzyme is given to them. This name was first given to them, or it was first originated from the study of the phage lambda. As you can see, this is the phage DNA, which is double-stranded. Phage attaches to the host cell and injects its DNA into the host cell, and this is the bacterial chromosome. So phage DNA, which is double-stranded DNA, and that DNA can be chopped up by enzymes. These are the restriction enzymes which chop up the DNA of the viral vector. It restricts the replication of the viral genome. Therefore, as it is restricting the replication of the viral genome, therefore these enzymes are known as restriction enzyme. There are various types of the restriction enzymes, and mainly restriction enzymes are categorized into three general groups. These include the type 1 restriction enzymes, type 2 restriction enzymes, and type 3 restriction enzymes. I will highlight the name as most commonly used are type 1, type 2, and type 3 restriction enzyme. The categorization of the restriction enzyme is based upon the composition of the enzyme, the requirement of the cofactor of the enzyme, nature of their target sequence, as well as the position of their DNA cleavage site, which is relative to the target sequence. So, mostly, as we have said, that there are three types of the restriction enzyme. Type 1 restriction enzymes are the complex endonucleases, and they recognize the DNA sequence, but they make cut in the DNA in random site that can be as far as 1000 base pair away from the recognition site. It means that they will recognize some other site, or they will cut at another site. They require several cofactors also, for instance ATP and magnesium ion, and these enzymes help in the methylation of the DNA sequences. And if I say that this is the DNA fragment and this is the recognition site where the type 1 enzyme will recognize, they will make a cut thousands of base pair away, or at other places, not at this one position. This was the recognition site, but they will cleave about 1000 base pair away, and their recognition sequence is 15 base pair long. They will recognize a 15 base pair sequence, and then they will make a cut in it. Then comes type 2 restriction enzymes. Type 2 restriction enzymes are remarkably stable, and they induce cleavage either within or outside the recognition sequence, but mostly they recognize and cut within the recognized sequence, so recognized site. So this is the example of the type 2, that it will recognize this sequence, and it will also make a cut at this sequence, and they are mostly 4 to 8 base pair. And these type 2 restriction enzymes are used in biotechnology, or we use these enzymes for the cloning purpose. Then comes the type 3 restriction endonucleases. These type 3 restriction endonucleases recognize a sequence, but they make a cut at different position, which is 25 base pair away from their recognition site. They also use the cofactors like ATP and magnesium ions, as well as they also required additional cofactors if they are involved in DNA methylation. So the type 2 restriction enzymes are most commonly used, and our focus will be on the type 2 restriction endonucleases, which recognize and make a cut in 4 to 8 base pair DNA fragment. Here you can see how the name of the restriction enzyme is derived, or we can say nomenclature of the restriction enzyme. They are named according to the name of the bacteria from which they are isolated. If we see the enzyme EcoR1, EcoR1 was isolated from E. Escherichia coli. So E is from the genus name, and C and O, it is from the species name. Then from which strain of the E. coli it was isolated? It was from the R strain, so it is named R, and the order in which it is isolated, so it is the first enzyme which was isolated from E. coli R strain, so it was named as EcoR1, and the recognition sequence of this enzyme is GAATTC. It means it recognizes this 6 base pair sequence, and it make a cut over here. It make a cut at this position. Similarly, here is the example of the HIN3 enzyme. HIN is isolated from Haemophilus influenza, and came from Haemophilus, IN came from influenza, and D came from the D strand, and this was the third enzyme which was isolated from Haemophilus influenza, therefore it was named as HIN3. Then comes the BAMH1, it is isolated from Bacillus amylus and from the strain H, and it was the first enzyme which was isolated, and these are the recognition and the cutting sites. It is the GGATGC, and it is the AAGCTT for the HIN3 enzyme. It is GGATCC because it will be the same sequence when we will read it on the other side. Here you can see the example how restriction enzymes work. That this is a DNA sequence which contain a cutting site. For instance, this is the cutting site of the EcoR1. This is the restriction site, and same is the recognition site for the type 2 restriction enzymes which recognize the 4 to 8 base pair sequence, and EcoR1 recognize the 6 base pair sequence, and it make a cut after G, and it will make a cut after G in both strands. So restriction enzyme will cut the sugar phosphate backbone, and then sticky ends will be produced, as you can see in it that these are the sticky ends. Now we want to introduce a DNA fragment. We have also treated the DNA fragment with the same enzyme, which is the sticky end. They have produced the sticky ends. Now this is the one possible confirmation that this enzyme can come and join over here, and then DNA ligase will seal these ends, will make a phosphodiester bond between these nucleotides, and in this way we will get a recombinant DNA molecule. As we discussed about the enzymes, that specific sites, restriction sites, and the specific sequence is the same, and mostly enzymes are 4 to 6 base pair. Palindromic sequences, here is the illustration of the palindromic sequence of the EcoR1. We call a sequence as a palindromic sequence when the read in the 5' to 3' direction, the sequence on the top strand is identical to the sequence in the bottom strand. So if you see the sequence of this from 5' to 3', and it will be same, which is GAATTC, and if you study this sequence, so read this sequence from the opposite end, which is 5 to 3 on the opposite strand, it is GAATTC. Such sequences are known as the palindromic sequences, and when restriction enzyme make a cut over here, and after this G, and after this G, then overhangs are produced. These sequences are known as the overhangs. So these overhangs are produced. It means there is the formation of two fragments, which are fragment 1 as well as the fragment 2. So these two fragments are formed with the overhangs, which are created by cutting with the restriction enzymes. What kind of cut restriction enzymes make, and what kind of overhangs are produced by the restriction enzyme? This is known as the mode of action of restriction enzyme. So mode of action of restriction enzyme is that it produces three kinds of ends. One of the ends are known as the 5' overhangs. 5' overhangs means the enzyme will cut asymmetrically within the recognition sequence. This yellow region which is highlighted, it is the recognition sequence of the enzyme. The 5' overhang will be produced when the enzyme will make cut in a site in such a way that a short single-stranded fragment extends from the 5' end, as it is only making a cut after the G. So this short strand will appear at the 5' end, and same on the complementary strand, it will cut after the G. So this short strand will appear after this. So this is known as the formation of the 5' overhang. Like if this is the DNA fragment and it has produced a cut over here and cut over here, then this fragment and then this long fragment, and then there will be a long fragment and a short fragment. So this is on the 5' side short fragment. This is also on the 5' and short fragment. Therefore, this is known as the 5' overhang. Similar to this 5' overhang, 3' overhangs are also produced by some enzymes. An example of 5' overhang is BAM-H1. That BAM-H1 will cut in such a way that it will produce the 5' overhang, while the 3' overhangs are produced when the cutting occur in such a way that the single-stranded overhang from the two 3' ends are shorter. Like if we see the example of this enzyme, which is KPN1 enzyme, it will cut at the end, like at the 3' end. Now the shorter fragment will be produced at the 3' end. Therefore, we will call it as the 3' overhang. There are certain enzymes which produce another kind of the cuts or the molecules which are known as the production of the blunts or the blunt end, which is provided by the small one. These are the enzymes that cut at precisely opposite sides in the two strands of the DNA without overhang. Like the recognition site for the small one is CCCGGG. It is CCCGGG. It will make a cut in the center, and such kind of ends are produced which are known as the blunt ends. So these are the blunt ends. These are the 3' overhangs, and these are the 5' overhangs, as this 5' end is smaller and here its 3' end is smaller. So it is known as the 3' overhang, it is known as the 5' overhang, and it is known as the blunt end. These are the examples of the restriction enzymes along with their cutting sequence or cutting restriction sites. Arrow indicates the phosphodiester bond where the cleavage will occur with the help of restriction endonucleases enzyme. And these steric indicate that these are methylated by the corresponding methylase in case of bacteria. In this, you can see the 4 nucleotides which are GATC, and if you see any other nucleotide like N, it means there could be G, C, A or T, and designate any nucleotide. We have studied about the naming of the restriction enzymes. We have studied about the types of the overhangs or ends which are produced by the enzymes. For example, BAMH1 will produce the overhangs, or these overhangs are also known as the sticky ends. But there are also examples which produce the blunt ends, like it is the ECO R5, or it is the HA3, or it is the PVU2, or these are the enzymes, other enzymes which produce the 5' overhangs or the 3' overhang.
This is the web page or website about to get the information about the restriction enzymes, because um, restriction enzymes which you are going to use, you need to select those enzymes based upon the nature or composition of your sequence, and what kind of experiments, or what is the purpose of your study. Why do you want to clone the gene? Either you want to clone the gene to make its multiple copies, or your purpose is to express it in the host organism or host genome, and then you want to purify the protein. So depending upon the purpose of your study, you select the vector and what restriction enzymes you will use. You can get information from the cloning vector. Cloning vector has a specific site at which the different cloning enzymes are present. This site is known as the MCS site or multiple cloning site, and use search those enzymes which are present in the multiple cloning site in your DNA sequence. If the enzyme make a cut in your DNA sequence within the gene, it means you cannot use that restriction enzyme at the 5' or 3' site, because if you use that enzyme, then instead of obtaining a single gene fragment after restriction endonuclease treatment, you will get two fragments and your whole gene will not be cloned into the vector. So you need to be very precise and specific in choosing or selecting the restriction enzymes. And restriction enzymes are mostly commercially produced by different labs. Here a link is given of New England Biolab, which is abbreviated as NEB. So if you order any enzyme from the New England Biolab, you can get information from their website. It will tell you about the sequence of the enzyme at which the enzyme will make a cut, and which overhang will be produced after restriction, and it will also tell you about various properties of the enzyme. For example, 37 degrees shows the incubation temperature for the AAT2 enzyme. That AAT2 enzyme shows optimum activity at 37 degrees centigrade. Then cut smart is a smart buffer which is supplied along with the enzyme. It means that the smart buffer is provided with the enzyme to be included in the reaction, and this buffer will provide all the optimum conditions to the enzymes to perform its function in vitro conditions. Then here you see the clock symbol which shows the time saver, that these enzymes can complete their reaction in within 30 minutes or 15 minutes. Then the next symbol shows that the enzyme can be inactivated by heat. For example, if you have added the enzyme in your reaction and after 15 minutes you want to inactivate the enzyme, then you just heat the test tube to 60 degrees centigrade or 70 degrees centigrade. Then the enzyme will become inactivated. CpG shows that it may be sensitive to methylation. Like if C or G is present, like you can see over here in its sequence, C and G is present, it means that it can be methylated at this position. Then R symbol shows that this enzyme is produced by using the recombinant DNA technology, and this butterfly symbol is for NEB, that this enzyme is synthesized in New England Biolabs.
Here you can see the restriction map of plasmid vector, which is PUC18 or PUC19. The size of the vector is 2686 base pair. And here is the sequence of the multiple cloning site in the LAC-Z gene. And in addition to multiple cloning site, it also contains some other restriction sites that we do not use for cloning purpose. We will use the sites which are located in the multiple cloning site, and we make a cut with the help of the enzyme, and if it disrupt the LAC-Z gene and our fragment is inserted in it, then the blue color colonies will not appear, as we have discussed in our previous lecture. So this is an overview how the restriction enzymes work. Like we have a plasmid and we have a DNA fragment. This purple color is the DNA fragment that need to be inserted into the vector. We will cut it with eco R1, and when it is treated with the eco R1, such sticky ends are produced, and this is the match of the sticky end with this, and the other, these are the mismatches. They will not incorporated into the plasmid vector. So we will treat the plasmid as such also with the eco R1. We get the sticky ends when the DNA recombination process occur. With the help of DNA ligase enzyme, we will ligate this fragment into the vector, and in this way we get the recombinant vector. While the mismatch of the vectors, like if there is no cut in the vector and vector is intact, there will be no insertion of the fragment. So in this way we will get the recombinant vector. So dear students, this was all about today's lecture, in which we started first with the types of the host which can be used for cloning purpose, and we discussed about the E. coli, Bacillus as subtilis, in case of the eukaryotic gene we can use the Saccharomyces cerevisiae. Then we have seen that which properties we should keep in our mind when we are choosing about the vector, that we need to understand the type of the gene that we are going to work, or what restriction sites we are using, what is the insert size or the size of the gene, and we need to know about the copy number of the plasmid. Then we move towards the restriction endonucleases, that these enzymes are isolated from the bacteria. Bacteria are the natural source of these enzymes because these are the defensive enzymes which restrict the growth of the viruses within the bacteria. Therefore, these are known as the restriction enzymes. Then we discussed about the different classes of the restriction enzymes, and we said that the type 2 restriction enzymes are used most commonly because they cut and they recognize and make a cut in the same DNA sequence. Then how much fragment they are produced, what kind of fragments are produced by these enzymes. We have seen that they produce the 5' overhang, 3' overhangs, or the blunt ends are produced, and the overhang which are produced, they are known as the sticky ends. And then these sticky ends helps to be ligated into the vector, which is also treated with the same enzyme. So this was all about today's lecture. If you have any question, please feel free to ask. Thank you.